on Monday for the lab that we're missing today, so you guys don't get shortchanged on being able to spend time with the models as we're really getting more into bones next week, okay? So you have a lab that's due Friday, I believe, isn't it? Didn't we set it up?
Yes.
Friday. So, and if anybody needs some extra time because of, you know, all the chaos and stuff of life, let me know on that. Was there anything else we needed to go over? Go ahead.
On the syllabus for tonight, they said we supposed to be going over PBL 1A and PBL 1B.
Yeah, ignore that for right now. I have to restructure because of the way class went tonight, that I'm going to restructure the whole syllabus. And we might be restructuring the whole syllabus a couple of times. I assume we'll be back in class on Monday, and God knows if we're going to get plucked out of class again at any point going along the way here. So sorry about that, guys, but it's the rules we have to live by, and we all have to, you know, inform them when we've had any potential exposure. So I'll modify the syllabus. I'm going to work. I have to modify the syllabus for both here and my Washtenaw class. So I'm going to be working on that the next couple of days, and I'll send you an email when I'm done with it and tell you about that. But we won't be taking a test, you know, in this week or next week. It'll be the week after that probably is when we're going to aim for our first PBL. Okay.
Now, how long do you give us for PBLs? Is it a week?
I give you not usually a week, but I usually give you like 4 or 5 days to do.
Okay.
Yet no one has ever complained that I don't give you enough time on this stuff. Like I said, I don't want you to be under the pressure of time. I'd rather you take the time to learn it. So if when I assign a PBL and you guys find you need more time as the due date's coming due, all you have to do is send me a little note, and I usually extend it for you. I just don't want to extend it too much because I don't want us to get real behind in our trekking through the class. Okay, but I want to kind of go with the flow of the class and the organic rhythms of the class as we're playing kind of fly by the seat of our pants here as to where we're going to be and what we're going to do. Okay. All right.
What time is the lab that's due Friday? What time do we need to have it in by?
I can't remember what I put down there. I think when
I think you said 5, but I just wanted to make sure.
Okay. If it's 5, it might be 5. If it's not 11 o'clock at night, I'll change it all to 11 o'clock at night to give you time. Okay.
Okay.
All right. And then we'll kind of go over if at the end, if you guys are having any problems with the membrane transport. I was hoping to get into that today. So we'll see if we make it that far. All right. All right. Any other questions? Any questions in class? Okay, you're here. All right, good. All right. So we've been doing all the chemistry stuff. Now we're getting to the part of that we are actually making the molecules that we need to make. All right. So we're going to go through the 4 major groups of organic compounds that you need to know: the lipids, the carbohydrates, the proteins, and the nucleic acids. So we're going to have to go through carbohydrates, what their function is, how they're formed, the different lipids, what their composition is, what their functions are, how they're formed, the proteins, what their function is, how they're formed, bonds. And in all of those, we're going to look at what are called the monomeric units, which are the basic units. Like a carbohydrate is made up of simple sugars that are linked one to the other. One sugar linked to another forms a disaccharide. You'll see as we get to it, I'll point out the monomeric units. All right, and then we'll look at the protein structure and how protein structure relates to the protein function. All right, and of course, we'll look at specifically when we're getting into our nucleic acids, we're going to have to look at DNA versus RNA, how RNA is different from DNA and how they function differently. And we'll also, as we're looking in at our nucleic acids, also describe and look at ATP. I know somebody last week, I forgot who, told me, oh, ATP. I didn't really understand that last time around. So hopefully we'll get you to understanding it a little bit better.
It was me.
Okay. It was Carrie? Okay.
Yeah.
Right. So the relationship between all of these, and then we'll start kind of showing a little bit how maybe deficiencies or abnormalities, maybe you form too much of a protein, for example, can become a problem for you. All right, so we talked before about organic molecules, and somebody said that a professor once told them that the definition for organic molecules is basically based on how much hydrogen there is in the compound. And that's not true. Organic chemistry is the chemistry of basically carbon compounds, by and large. There's some other compounds in there that don't contain carbon that we look at, like urea for organic chemistry, but it's by and large the chemistry of carbon compounds. And it's largely carbons combined with other carbons, particularly hydrogens, oxygens, and nitrogens, some other things thrown in here and there. And this is because carbon, with those 4 electrons in its outer valence, has a unique ability to bond to itself in long chains over and over and over again. This is called catenation. Okay, chemical catenation is when you link the atoms that are the same atoms, one carbon to the next carbon to the next carbon to the next carbon, and then basically we're going to be saturating it with hydrogens. Now, this definition might feel a little disorganized or nebulous or not really well defined, however you want to describe it, and that's because that's true. There's a lot of argument amongst chemists as to what defines specifically organic chemistry. So don't feel too unique if you feel like you don't have a really good grasp on that. What I'll say is, after a while, you start looking at all these molecules, you start getting a feel for what is an organic molecule. And thankfully, we're not in inorganic or organic chemistry, where you're probably going to get pigeonholed into defining it more narrowly. Okay. All right. So when we start concerning ourselves with organic molecules, the sort of main molecule that we center around with organic molecules are carbohydrates. And that's because of that catenation of the carbon being bound to other carbons. And on these long carbon chains, where the carbon's bound to a bunch of other carbons, we'll find that there will be thematic functional groups that we see over and over and over again. And these are often called organelle groups or functional groups, or you'll see them abbreviated as R groups. And you know what? I'm going to get up and I'm going to turn the light on and walk as I talk here, because I see it's getting dark in here. All right, so R groups are these functional groups. They're going to be groups that make your chemical either a alcohol, an acid, an aldehyde, a ketone, many of these other compounds that you've probably heard of over time. I'm even going to bring another lamp of mine. See how unprepared I am for us going, what do you call it? Going virtual today. All right, try and get as much light in here so that you guys can see me and all my great glory and beauty at home. The one beautiful thing about being at home is like I can wear like my sweat jacket that I wouldn't walk into school live with. So I find that when you're more comfortable, you think better, So I often wonder why we have such a obsession with needing to dress up and such when we go to work. Like when you want to do work around the house at home, don't you just want to be comfortable in your tennis shoes, nice comfortable sweats and stuff? Isn't that like the one thing that all of us have liked about being at home through COVID is kind of more comfortable all the time, right? Maybe we're too comfortable because now we all have to wear elastic waistbands. All right. So our organelle groups, Nat, we'll go through that now.
Jessica,
so the organelle groups are also known as functional groups. And as I said,
it's not homework, but she said we don't have to do it, but I need to do it so that I need to practice the math.
somebody needs to turn off their microphone. Your child, I can hear yelling, I think at a dog or something. Okay. All right. So you guys can hear me and turn off your microphones. All right. So functional groups for alcohols mean that you simply have an OH hanging off of your carbon main structure. The smallest alcohol is when you have just one carbon and an OH group, and that's methanol. And when we build up to ethanol, which is what you all want to be doing after this class, right, is dipping into the Tito's, that is ethanol right here. Okay, so that's two carbons and an OH group, and you see all the rest is surrounded by hydrogens. So methanol is what is like the side product when people are making moonshine, and this is what they die of when they drink it. Just one carbon different, right, from methanol to ethanol. This is toxic to you, this is not. The third is isopropanol or isopropyl alcohol, what you use to swab off your cut or swab off and sterilize before, you know, sticking a syringe into somebody. Okay. Now, if you were in organic chemistry class, you'd have to learn these stems and build off of them. We don't have to do that in here. I just want you to recognize, be able to recognize that an OH group means you're dealing with an alcohol. And the alcohol with oxygen on here with its two lone pairs of electron makes this molecule a little bit polar at the end where the alcohol is, okay? A little bit polar. All right, other functional groups. So we have the alcohol. If instead we lose we'll lose a carbon being bound to a hydrogen, and instead we double bind it to an oxygen, and then it has another hydrogen on it, then that end is called an aldehyde. These are things like formaldehyde that you use to preserve a cadaver or specimens in your lab work. Glutaraldehyde, okay? Ketones are when you lose the hydrogens and have a double bonded O in the middle of the molecule. This R and this R on it just means all the rest of the carbons, all the rest of the carbons in the big long chain of carbons. And an amino group, instead of adding an OH or OH type structure on, we're adding an NH or specifically an NH2, okay? And this is going to be the basic structure of an amino acid. So an amino acid will have an amino when we see it at one end and an acid group at the other end. All right, esters, we won't deal with too much, but that means your carbon is double bonded to one oxygen and single bonded over to another one, okay? In ether, you have a carbon that is all single bonded, connected to one O, which is connected to another carbon that's all single bonded, okay? So single bonds around that oxygen. Sulfhydryl is just an alcohol group that instead of having an OH, you have an SH, okay? So you have a sulfur there, which is just below oxygen in the periodic chart. So they behave very similarly. Carboxyl, this is going to make things into carboxylic acids. This is the acid end of a molecule. So when you see an alcohol group and an O group like this, is called a COOH, or that makes everything acidic, okay? This, you lose this proton. Typically when you put this in water, this is highly polar at the end because you have a double bonded oxygen out here with its two lone electron pairs, and you have this alcohol group that loses its proton and it's got a single oxygen, but it has its two lone electron pairs. So this will become very acidic in water. And then we have a phosphate group. This is the one that only one we're looking at that doesn't have a carbon in it. And so that we'll have our long carbon chains down here, and then we'll have this phosphorus surrounded by one oxygen that's double bonded and three oxygens that are all single bonded to the phosphorus. All right. Okay. And you don't have to memorize the structures too deeply here, but kind of really be able to recognize what these are as we go through it. And we'll go through it enough that hopefully you'll start being able to recognize the groups. Okay, so like I said, we're going to spend a lot of time talking about amino acids because they're the building blocks or the monomeric units for our proteins. And so all of our proteins are one amino acid hooked to another amino acid hooked to another amino acid. So you have your amino group at one end and your carboxyl or carboxylic acid group at the other end. So that's amino and that's acid right there. All right. So all of our small organic molecules that we've been looking at are going to combine into larger macromolecules. And as I said, this is because of that catenation, which is carbon's miracle ability to bond to itself over and over again, making long hydrocarbon chains. All right. The macromolecules will be also very repetitive. So they will be repeat units of monomers, and the monomers for carbohydrates will be sugars. So we'll take simple sugars, we'll put two of them together, which will be disaccharides. And then we'll build them up into bigger units, which will be oligosaccharides till even bigger till we're at polysaccharides. And at some point we start calling them carbohydrates, okay? Whenever we join our little monomeric units together, kind of knitting them together along the way, we're going to form new bonds. And as we talked last time, we talked about synthesis reactions and decomposition reactions. And we use water to help build our bonds together, to condense two molecules together. So we call them condensation reactions. When we use water to put two molecules together, you'll see that in a minute, okay? And we call it hydrolysis when we use water to split a big molecule like a carbohydrate down into its component parts like simple sugars, okay? All right, so just to remind ourselves, when we did a dehydration synthesis, we took A and B and we put it in water. So typically I would write up here water above the arrow, and we form compound AB. This is called a condensation reaction, okay? Dehydration synthesis is sort of the opposite, okay? And here we split our AB back into OH. I'm sorry, dehydration synthesis is the same as a condensation reaction. It's hydrolysis, the decomposition, that's the opposite. I misspoke. So we're going to take our A that has an OH group on the end and B that has an H. You can see that that's the same thing as if I just wrote water above here, because this is OH and H is H2O. And they unite then to form a new bond, kind of swapping partners here, where this becomes A and B together, and then OH and H are together. So then you get water as your byproduct. So you've condensed it. And if you think about this, this is what condensation is, right? The water collects on the condensation in your window. When you go to buy condensed orange juice and you want to make it into orange juice, you have to add the water to it, right? It's condensed. The water has been removed from the orange juice, right? If this were orange juice plus water. So we're always going to want to add the water back into the orange juice to make it, you know, into its new product. All right, and you can see here, this would be A. So we have C with its functional groups over here. This is actually an amino acid right here, condensing with another amino acid. So here's the amino ends, here's the acid end. And typically in a protein, when we're going to knit our molecules together, the acid end of one amino acid reacts with the amino end of the next amino acid. And we're going to lose water right here and form a new bond between this carbon and the nitrogen. And you'll see over here, that indeed is what happens. All this looks the same right here. And then we have our carbon, we have that OH drop off, and it hooks on over here to the NH. So you just have NH here. And then you have the rest of that molecule over here, and OH is off to the side now. We've removed the water from it. All right. All right. So when we do this with carbohydrates, and I'll show you a carbohydrate polymerization from the monomers in a second. Carbohydrates are going to be our main organic substances that we use for cellular energy. It's what you use for food. Even if you like want to run a marathon or something, pretty typically you carb load to supply you with immediate energy before you go and run your race. All right. Carbohydrates contain carbon, hydrogen, oxygen, and sometimes they can contain nitrogen and phosphorus and sulfur and other substances, but C, H, and O are the main three substances always to make something a carbohydrate. And the ratio of hydrogen to oxygen is very close. It's almost always 2 to 1. So you can see here, C6H12O6. You have 2 hydrogens for every oxygen. This is the formula for glucose. It's also the same formula for all the what we call isomers of glucose, and we'll get to what that is in a second. All right. So when we first classify our simple carbohydrates, the most simple ones are called simple sugars or monosaccharides. These are things like glucose and fructose. All right. When we put 2 sugars together, like if we hook together a glucose and a fructose, we make sucrose. Okay. So disaccharides, 2 monomeric units combined. And then we can do this reaction over and over and over again until we get long polysaccharides. And these are our complex carbohydrates. This is what builds up starch, okay, like in your potato or in your bread, glycogen, which is the storage form in your liver, or cellulose, which is the way plants store carbohydrates. So this would be things like, you know, your celery or something. All that roughage and fiber in there is cellulose. All right. So again, carbohydrates, commonly called sugars or starches, the monomeric unit or basic unit from which we build all carbohydrates are called monosaccharides. We can build them into disaccharides, and kind of a stage in between disaccharides and polysaccharides are oligosaccharides, meaning more than 2, but probably less than half a dozen. So probably like 3, 4, or 5. All right. And all of these will be starches. We'll have C, H, and O, roughly with the formula of CH2O1, okay? Or CH2O to the N, multiple numbers of times, okay? All right. Now, before I go on from all that, do you guys get this, how this formula is written? N out here. So if we multiply by 1, that would be C1H2O1. If we multiply by 2, that would be C2H4O2. If we multiply 3, it would be C3H6O3. Okay. So you're always saying, I always have twice as many hydrogens as I do oxygens, and usually also carbons. All right. But it tends to be the ratio of the hydrogens to the oxygens that kind of stays a little bit more consistent. All right, so carbohydrates have isomeric forms. This means you have the same chemical formula, but you can structure it differently. So when we say our typical glucose of C6H12O6, we can see that right here. And this is how it's arranged, with the double bonded O out here forming a aldehyde at the one end, and then you have all these alcohol groups going down on each other side of the carbon. All right, galactose right here also has the same formula. If you count it up, it's still C6, it's still H12 and O6. And the big difference is all this looks the same, all this looks the same, this looks the same, but right here, you flip which side of the carbon the OH is on, and that does end up making a difference or matters. Fructose also has 1, 2, 3, 4, 5, 6 carbons. And if you count, there'll be 12 hydrogens and 6 oxygen. So it's an isomer as well, but its carbon that's double bonded to the oxygen is in the interior of the molecule from glucose. But other than that, it looks the same as glucose, right? It has the OH on this side of that same 4th carbon right there, okay? All right, and if we wrote these out, we sometimes write them in their open form, and sometimes you, see these structures written in their, what we call their closed ring conformations. So this would be where we would say this carbon comes and what we call attacks or polymerizes, because as all these carbons kind of twist and bend around their structures, they start bending around so that they're at 6 carbons right back up here, leaning toward the beginning of this molecule. And so sometimes you reform a bond between this carbon and this carbon, and you're kind of throwing out this extra carbon kind of like hanging out here with this alcohol group. You see? Fructose, because it's carbon that's attached to the double bonded oxygen, is in the interior of the molecule over here. You see that it's going to attack kind of, let's pretend it's down here. Instead of attacking up here, it would attack and be drawn to the carbon that's double bonded to that oxygen because it's so electronegative, and it forms its ring structure as a pentose, okay, instead of a hexose. 6 carbons in the ring here, 5 carbons in the ring, but you have an extra carbon hanging out over here as well as over here. All right, so our monosaccharides are isomers of each other. They have the same formula, same number of carbons, hydrogens, and oxygens, and they're all the simple sugars, and they have from 3 to 7 carbons in them. Most of them that we focus on are the 6 carbon sugars. And glucose and deoxyribose are also examples of common monosaccharides that we will see when we go to build our DNA and our RNA, okay? So they look a lot like these, and we'll show you how they look different later on. All right, so disaccharides, like I said, are formed by a condensation reaction. We're going to put the 2 molecules together and form a new bond and remove some water from it, okay? Then new disaccharide, the reaction in reverse is called hydrolysis, which is a decompensation using water to split it. So if we go in this direction and we have a glucose and a fructose, what's going to happen is you'll find this end of the fructose and this end of the glucose together are going to kind of move next to each other. And as we said, in solution and everything in our bodies, we kind of drop off a lot of these H's as protons, and we're going to form a bond here, and we're going to remove some water. So you'll see we'll remove this OH group and this H group, and this carbon right here is going to bond over to this oxygen right here. And you see that happens. Here's our carbon still with the hydrogen on it, and it bonds right over here with that oxygen right there being the linkage, okay? So you can kind of see how that carbon's coming over here. If you drew an arrow, it's attacking this oxygen here with its 2 lone pair electrons. It's very drawn to it. And so then we have carbon over here, bond to oxygen, and this oxygen stays bound to this carbon, but we kind of draw it with a new little loop-de-loop look right there. So that's sucrose, okay? Our glucose and our fructose, and we removed water. Oops, and I should say, in the reverse, of course, is using that water to come in here and break apart the bond that we just formed and go in the reverse direction. And we hydrogenate this back here to an alcohol group, and we put the alcohol group back on here, hydroxylating that glucose, split apart. Okay, formation of sucrose again. If you want to see it, kind of draw it out here. We're removing the water away, and we're going to have a new bond form between the oxygen here and the and over here to the carbon. So it can kind of form, you know, either off of this OH or this OH in a little bit different way. Whether it plucks off the hydrogen of one and the oxygen of the other, doesn't matter which one you pick. It ends up looking the same, okay? All right, so if you do that same condensation reaction, condensing the two together, removing water over and over and over again, you start linking together glucose to glucose to glucose to glucose. And that's how you form a polysaccharide many times over, taking the hydrogen out of these and doing that reaction through the dehydration synthesis. And this is how you form straight long chains, or ultimately even branched chains that become the starch in our body, okay? Starch, cellulose in plants, all these are polymers of glucose, okay? And starch is going to be highly branched, easily stored in your liver. When you look at the structure of glucose, we can draw it so many ways. You got to kind of get a little bit used to seeing it. You're probably going to see it most often being drawn in its ring form like this. And there's even a shorthand for this where they leave all the H's off. Okay. So you can have it just kind of written out open chain, ring form. And here you'll see structural models of glucose. Here again, looking at it, you can see the isomer here, main isomer for glucose, all right? All that straight carbon atom. Here, they bend around again and form the ring structures, and this is the way you'll probably most often see it, is without all these extra hydrogen groups and things just kind of removed, and you recognize that as glucose. If you see a little circle drawn in here, then that means every other thing is double bonded and it's no longer a glucose molecule. You're dealing with benzene then. So monosaccharides, disaccharides, joined over and over again, sometimes branching, you know, off of different carbons, all right? And you end up with a long chain of polysaccharides. And you can see we have multiple organic molecules made up of its monomeric component parts that we'll see over and over again. Carbohydrates, lipids, proteins all folded back on each other, and of course the big alpha helix structure of our DNA. So that's our nucleic acids. And we know from before, you've probably learned your lipids has the phospho head that's highly polar, and then you have the non-polar fatty acid tails, okay, which we'll discuss right now. All right, so lipids are the primary components of our cell membrane. The whole thing, why we need these lipids, these phospholipid bilayers, is so we can compartmentalize everything, all right? You compartmentalize so that the fluid on the inside of your cell is separate from the fluid outside of your cell, and they have to exchange things quite often through membrane channels and have routes of communication for exchanging materials. And we'll spend some time on that. But the lipids help us maintain our different integrities of our interiors of our cell versus the outside of our cells. And even within our cells, we maintain the integrity of the cytosol as being different from what's inside the nucleus. The nucleus is bound by a phospholipid bilayer as well, the nuclear membrane. Things like your mitochondria is also bound by a phospholipid bilayer. And so the interior of the mitochondria is different from the rest of the cytosol. So that same type of exchanging through some channels and carrier proteins, things like that, will go on intracellularly as well. All of our lipids are going to be made of carbon, hydrogen, and oxygen, but it's going to be in kind of a different structure and numeric relationship than what you found in carbohydrates. Carbohydrates, remember, your oxygen and your carbon tend to be about the same number, and your hydrogen is twice as many, all right? And that's why I think where Anna was told that it's, you define organic based on how many hydrogens there are. I think it's talking about carbohydrates, and we always have usually twice as many hydrogens as there are carbons and oxygens, but it's not the defining thing for what is a organic molecule, because for instance, lipids are organic molecules as well, and you'll see that ratio switches. So our lipids, we know, need to exclude the fluid components, separate from each other, so they can't really dissolve in water. They're going to be nonpolar. They're insoluble in water. Water is repelled by them, okay? Water is repelled by the lipids. Lipids are what we call hydrophobic, okay? They don't love water. They fear water. All right, so these two, our lipids, will also be long chains of hydrocarbons.
I didn't get that.
Oh God, my other computer's going on, sorry. Sometimes I really hate Siri. So, I think Siri heard that. All right, long chains of carbon and hydrogen. Hydrophobic, so all the things that you think of like fats, oils, even waxes. Waxes are a particular type of fatty substance. They're all in soluble water. They all allow us to compartmentalize. They have a much higher oxygen content than do carbohydrates. And so if you're replacing and putting more oxygen content into the carbohydrates, you're typically also removing some of the hydrogens, all right? Fats will provide twice as much energy as what carbohydrates do, okay, in terms of kilocalories. And you know this if anybody is dieted. Fats always have more calories than carbohydrates and proteins. And our fats, we're going to talk about three basic types of fats: the triglycerides, the phospholipids, and the steroids. You will see that the triglycerides are our main storage form of fat. So when you got fat on your thigh, fat in your butt, fat in your belly, all the fat that's filling up your adipose cells that we love so much, those are your triglycerides, okay? The phospholipids are what are going to form the membranes of every single cell specifically. And then steroids will be derived from cholesterol. Cholesterol is also inserted in our cellular membranes, but the steroids, cholesterol will be the precursor molecule from which we make all of our steroids. And our steroids are going to be things like our sex hormones, testosterone and estrogen, and some other sort of inflammatory molecules that we'll talk about in a little bit. All right, so all these fats, like I said, important components of the cell membranes. The most abundant, of course, is our triglycerides, because that's our storage form of fat in our body, all right? We use it for cellular energy. We call upon it to break down those storage forms of fat when we're not getting enough energy just from our food alone. And all this triglycerides, all the lipids in general, are going to contain carbon, hydrogen, and oxygen, but less oxygen in it than what's in the carbohydrate content, okay? Much lower oxygen. And when we build these, we'll see that when we build our triglycerides in particular, triglycerides are called three glycerides, because we're going to make what's called a glyceride bond, in which we're going to take a glycerol molecule and bind three long hydrocarbon chains of fatty acids to it. Okay, that's what defines a triglyceride. Remember that all of these are critically important. The lipids are really critically important for energy, providing structure, forming the integral parts of our membranes, also, making your functional steroid hormones that you need. So let's look first at what a fatty acid is before we bind it to the glycerol, okay? So a fatty acid is going to be any long carbon chain that has a carboxyl group at one end of it. So we're going to have carbon, with its carboxyl group CO, OH, O, OH, here's a C, here's a C, and R just means C after C after C after C, okay? Fatty acids can be what we call saturated with hydrogens, in which case we call them saturated fats, or if not all the carbons are surrounded by hydrogens, then we call it unsaturated because there's some double bonds. So if we only look at a fatty acid, if we look at a fatty acid and only see one double bond, we call it monounsaturated. And these are some of the healthiest oils, like olive oil or avocado oil. Polyunsaturated means there's two or more double bonds. And this is going to be some of the are not as healthy as our saturated oils, considered generally not as healthy. These are the key ones, the monounsaturated ones for your health. Okay, so let's look at what's saturated and unsaturated. So fatty acid, it has a carboxyl group, a carbon double bonded to an oxygen at the end of the molecule, and also singularly bonded to an alcohol group. So CO, OH, okay? The rest of the carbon chain, this would be what's called the R group down here, carbon after carbon after carbon. They're all saturated or surrounded completely by hydrogens. Every other bond, every carbon forms is with a hydrogen. An unsaturated fat, here we can look at it and say, once again, we have our carboxyl group down here, and then we have our long R group. But here we see not every carbon is completely surrounded by hydrogens. Instead, it's lost a couple of hydrogens and instead formed a double bond between this carbon and this carbon, and this carbon and this carbon. So this is diunsaturated or polyunsaturated, you could call it, because it's more than one. If we only had one double bond here, and this one had a carbon bound to a hydrogen and bound to another hydrogen here, it would be mono, one unsaturation point in this long thing. That would be like olive oil has that, okay? So saturated, unsaturated. And you will see that what matters, why these double bonds here, is they put, as you can see here, little bends or kinks in the molecule that affect the way that molecule reacts with other molecules. Those little bends or kinks, all right? Now, here we're going to start binding a fatty acid, a big long, this one, is this one saturated or unsaturated? It's saturated, right? It's saturated, so it tells you right here, but you can see every carbon is straight, a long straight chain, no double bonds, okay? So it's completely saturated. It has 15 carbons. Because it has 15 carbons, it's called palmitic oil, okay? Then we have its carboxyl group, and it's going to bind to glycerol. Glycerol is a molecule that has a carbon with two hydrogens and an alcohol. Has a carbon in the interior with only one hydrogen and an alcohol because it's bound to the carbons on the other side. And then the third carbon is also bound to two hydrogens and an alcohol. And so you can see when you go to bind this carboxyl group to this alcohol group, you can remove the water and then form the bond between this oxygen over to this carbon, okay? So you are doing, once again, a condensation reaction, also known as a dehydration synthesis. Synthesis because you've made a bigger molecule, dehydration because you've taken water out. Taken water out is another word for condensation as well, okay? All right, so we do this 3 times over on the molecule, and we bind our 3 fatty acids to 1 glycerol. We remove 3 waters from it. It's going to be enzymes in our body that are causing this dehydration to take place. And suddenly we form this new type of linkage right here, which will end up being called an esterified or ester linkage. I don't care so much that you know that, all right? But I'm just telling you in case you want to know. So you can see how all these fatty acids are bound onto glycerol. Is this a monounsaturated or polyunsaturated triglyceride? Or is it saturated?
Would that be a polysaccharated?
Yeah. Every single fatty acid is completely saturated with hydrogen. You don't see any double bonds because you don't count the double bond to the oxygen. Okay. All right. How about this one? Here's another triglyceride. What do we see here?
Is this one unsaturated?
It's unsaturated. Is it mono or polyunsaturated?
Poly.
Poly.
Count the double bonds. How many double bonds do you find in the fatty acids?
So it's
Just
mono because it's
one.
There you
Only
go.
one.
Yep. Because you see these two carbons here, but it's only one bond between them. And that's always tricky. So you got to look close. Okay. All right. Here, this is what that polysaturated looks like, or we just call it completely saturated. Okay. So nice and straight, no kinks. Here, when you put the space filling model in, you see with the unsaturated where you have double bonds, you get the kink. So you can see there's one double bond here. You can see there's another double bond here. You can see there's a double bond over here too. So at least 3 double bonds. Okay, let's look at this one again and let's go through everything. We have our glycerol portion of the molecule here. We have that ester linkage that if we put to split it back to make it into ester, to make it into glycerol and its component amino acids, these would all be OHs right here, and you would be turning these into COOHs right here at the end. All right, so you can see right here, you have one double bond and you have this completely saturated and this completely saturated. So this is again, a monounsaturated fat. So this is probably a very healthy fat. We also describe the configuration of the H's around the bond here when it's double bonded because it can twist differently. I should probably have drawn, I would normally draw this out on the chalkboard for you, but you can see the 2 hydrogens are here on the same side of the double bond. We call that a cis configuration or a cis unsaturated configuration. If this H were over on the other side, when the H's are on the opposite sides of the double bonds, then we call it trans. And you've probably heard about how bad trans fats are for you. Okay, so we like our cis fats a little bit better when this H is over here and it really twists the molecule up a lot more. Your body has a lot harder time dealing with that. So our trans unsaturated fatty acids are even worse for us. All right, so now we're going to build our.
I'm sorry, can I stop you for 1 minute? I was trying to the H atom. That I was trying to understand that part.
Yeah, so
trans.
let me try to go back here and I'll try to get out and draw it in. So let me go here to home, insert draw. Okay, I got to look back. I'll do it in red. So if we instead put our H over here, H, and we had it single bonded here, and we got rid of this, that would be the trans configuration, okay? Where the hydrogens are on the opposite side of the double bonds, okay? And that kind of twists and kinks it even a little bit more, all right? So that's what makes a trans fatty acid. Slideshow from current slide. All right. All right, so a lot of our complex lipids, now that we just hooked our fatty acids onto our glycerol to make triglycerides, okay? We're not dealing with phospholipids or steroids yet. We're just dealing with complex triglycerides, okay? Sometimes we can also add in other molecules complicating these things, as we do when we make our phospholipid bilayers. So we add phosphorus on. Sometimes we'll add some nitrogen, sometimes we'll add sulfur or something else. So all we've ever heard our whole lives growing up is our cell membranes are phospholipid bilayers. So we're going to look at closely at what that is. So we're going to have our glycerol hooked on to 2 fatty acids. And at the third area, instead of having a third or a triglyceride, we have it as a diglyceride bond. And then on the third carbon of the glycerol molecule, we're going to add a phosphate group. Now, remember our phosphate group was crazy. It looks like, let's draw again. We'll just draw it here. It's got a phosphorus surrounded by 4 oxygens, one of them being double bonded, okay? So this is super polar because you have all those oxygens that have all their lone pairs of electrons, okay? So these oxygens are going to be highly polar and they're always going to be trying to pull the electrons away from that phosphorus molecule. All right, so it's going to try to pull the electrons away, pull the electrons away, pull the electrons away. That makes that end of the molecule that has that phosphate on it super highly polar. So it dissolves readily in water, but a little bit more reactive. Okay, so that's going to be a very polar end of the molecule. All right, so as we get into all this, we're going to say those phosphates are highly polar. And the other ends that's going to have just all those fatty acids bound to it are going to be relatively nonpolar. Okay, and so we're going to find that that molecule twists around so that the phosphate heads are going to face the cell interior of the cytosol, where it is really polar on the inside, all that watery solvent, and also face the outside of the cell, the phosphate heads. Okay. And all the long tails of those fatty acids in the center are going to form up the center part of the phospholipid bilayer, okay? So this is what it ends up looking like. So normally we had a third carbon right here also being bound to a long fatty acid tail, but instead it's being bound to this highly polar phosphate group. Here you have a nice long saturated fatty acid. Here you have an monounsaturated fatty acid. And so we tend to draw this as the phosphate group, this part up here, that polar head. And then these are the 2 long tails. And then we line them up. They line up one after the other, all those hydrophobic tails in the interior, keeping water out. But then the hydrophilic part, the part that's really polar, likes to face the water of the outside of the cell and also likes to face the watery part of the inside of the cell. But you can see anything that wants to get through from the outside of the cell to the inside of the cell is going to have to somehow get through and make it through all this hydrophobic fatty part of the interior of your cell membrane. Okay. So here's a cell membrane that has lots of saturated fatty acid tails in here. Here's a cell membrane that's got a lot of kinks in it because it has a lot of unsaturated fatty acids. And here's one that I think has like a little mix. But you can see that in this last one where they have it here, you can see that there's some like sugar groups hanging off of some of these fatty acids. And we'll get to that sort of at the end of the lecture of how sometimes we glycosylate some of our phospholipids. Okay, we glycosylate a lot of our membranes. It's kind of the basis of, any of you know your A1C, your glycosylated hemoglobin for tracking diabetes. This is the idea of how much sugar sticks to the membrane of your red blood cells. Tells us how much your sugar level has been raised in your blood over the past 90 days. And if you didn't understand what I just said, don't worry, at some point you will, okay? All right, so phospholipids, we have our lipid head, okay? We have our water-loving phosphate group, okay? And then we have our water-fearing tails, our hydrocarbon tails, our fatty acid tails. And so we can draw it like this, drawing it all out, or we can draw it with the hydrophilic head and the hydrophobic tails. And here you see you have all your hydrophobic heads pointing to one part, say the outside of the cell. Here you have all the hydrophobic heads pointing to the inside of the cell, and all those fatty acids excluding exchange in between. That's what causes the barrier, is that part in the interior. All right. Here again is another picture of the fatty of a phospholipid molecule. You can see right here we have our phosphate group. Sometimes other things attach onto it, like we have a little nitrogen containing compound. Here again, you have your fatty acid tails. All right, and you see this, they drew one here that has a little kink in it right there, and this one is completely saturated, or drawing it that way. All right, so now let's go on to steroids. Steroids are a different part. So we looked at triglycerides, we looked at phospholipids. Now we're going to look at steroids. Steroids all have the same basic structure. They have these 4 carbon rings. 3 of them, A, B, and C, have 6 carbons, and D is a pentose that has 5 carbons. You can almost see where these out here would form like 6 carbon rings if you completed the circle, but they're incomplete. Cholesterol and steroids will both affect membrane fluidity. And they're kind of interesting in that they affect fluidity in both ways. They impart a certain level of structure and rigidity, but they also keep enough bending and flexibility in our membranes, you know, that would otherwise be a little bit stiff if everything just lined up like this on the phospholipid bilayer. Okay. Sometimes we lodge a cholesterol big clump in here in between, and it gives it some additional shape. All right. Where was I? Okay. All right. So our steroids have those 4 rings, 3 6 carbon rings, 1 5 carbon ring, and steroids are widely distributed throughout the body. You have your estrogen in your ovaries. Men have testosterone in their testes. Women have testosterone too. You know, you have a little bit when you're, you know, a certain amount when you're younger, and you become a little bit estrogen depleted and a little bit more testosterone heavy as you get older. And that's why in menopause, you start getting some of those, what we like to call our lovely manly traits of getting things like little whiskers and that as you get older and you get depleted of estrogen. But both testosterone and estrogen as steroids have this same basic structure to them. They're just a little bit different. They're involved. Cholesterol is necessary. We've kind of been trained to think of cholesterol as evil, but it is absolutely necessary for life. It's in every single cell membrane of your body. And some of it comes out of the membranes and is used, like I said, to synthesize things like your adrenal gland hormones and your sex hormones. Okay. So cholesterol is the main basic molecule from which all sex hormones and adrenal hormones are derived. Okay, so there's cholesterol once again. Look at cholesterol, look at estrogen, look at testosterone. Not much different is testosterone. You can see both have an OH here, except the OH faces away from us, and testosterone here, it's in our plane. That's what that dot, dot, dot means, that it's kind of going out toward the back. The methyl group, CH3, is the same. We have the 3 same carbon rings, except in testosterone, we only have this double bond and this double bonded ketone group here. Whereas in estrogen, we have what's called a benzene ring of double bonds all around the ring, and we have an alcohol group on it. So really not big differences, but you see how these little differences in the structure of these molecules give us such sharply different effects of what testosterone domination in your body will do versus estrogen domination in your body. And we'll talk about that more when we get into sex steroids next semester. All right, so your steroids and your prostaglandins. Here's another term, and this is a nebulous term students have hard times with because they're really kind of a misnomer. They were first hormones that were found in the prostate gland. So that's where they're given the name prostaglandin, prostate gland, okay? But they really are more like inflammatory hormone molecules that are throughout your body, okay? Just first discovered in the prostate. And they too are derived from that steroid nucleus, all right? And so you will see as we go through the semester that a lot of the synthetic hormones that we use that have therapeutic value will also include a large number of these sort of anti-inflammatory agents that are based off of prostaglandins, okay? So they're all derived from a substance, a particular fatty acid called arachidonic acid, which when we're forming those triglycerides in our membranes, they have 20 carbons, they have 5 rings on them. And when they come out of the membrane at different times and different points, they're released from the membranes in response to different stimuluses, okay? They're called also tissue hormones. And like I said, they could be pro-inflammatory. When we're taking things like a COX-2 inhibitor like Celebrex or an NSAID like Anaprox or naproxen sodium, things like that, we're trying to stop the release of these prostaglandin molecules from our cell membranes, because those molecules, when they're released, cause inflammation, that terrible, uncomfortable reaction where you have rubor tumor calor dolor, or redness, heat, swelling, right? And pain throughout your body, okay? So that's what prostaglandins do. So here's a prostaglandin, and you can see it's kind of like that basic steroid nucleus. It has that 1 5-carbon ring, and here it has 1, 2, 3, right there, incomplete 6-carbon rings, okay? But they kind of line up just like they're going to almost form a ring. All right, and then moving on from our lipids, so we did steroids, we did triglycerides, we went over fatty acids, and we went over phospholipids. Lipids don't really have a monomeric unit. Like all carbohydrates are built up from those simple sugars hooked together. Lipids don't really have a monomeric unit, because you can't say fatty acids because steroids don't have fatty acids, right? Our phospholipids kind of have fatty acids, but they have the phospho on the end. And our triglycerides are definitely 3 fatty acids and a glycerol. So we can't really say lipids have one monomeric unit. Now, proteins also have a monomeric unit, which will be the amino acid as we get to it. Proteins are our most abundant and important organic molecules. They form the basic structure of our body. Like when you look at your muscles, okay, that's going to be protein. You know that when you want to eat a protein-based meal, you go for meat, which are the muscles of animals, all right? Proteins are made from carbon, hydrogen, and oxygen. And the thing that characterizes them, instead of looking at how much hydrogen and oxygen like we did with fats and with our carbohydrates, we see the nitrogen in the molecule, and you're immediately clued in on, is this an amino acid? If it's an amino acid, we're making proteins likely, okay? So amino has the amino end, the NH2 end, and the acid end, the COOH, where C is double bonded to one O, and then is also single bonded to an OH hydroxyl group. All right, so amino acids are the basic building blocks. We have 21 basic natural occurring amino acids that our body uses. 8 of them are what we call essential amino acids. They're not made by the body, so we have to obtain them from outside sources, meaning your diet, okay? The non-essential amino acids are synthesized de novo or synthesized newly, meaning de novo, by your body. You can make it from other things that exist in your body. All right, so proteins, like I said, form the basic structures, the structure of our cells. You know, we have a lot of proteinaceous component parts, and they will be the major components of all of our tissues, our organs, particularly our muscles. And proteins will be defined by their unique shapes. They form a lot of our functional molecules like enzymes and hormones, okay? A lot of hormones that communicate with each other. And not necessarily our steroid hormones that are fat, that are our sex hormones, okay? But a lot of the other hormones that communicate throughout your body that we'll get to also next semester. All right, so a lot of them will be these communication molecules, hormones and enzymes, and the enzymes in particular will fit like a lock and key, so the shape of the protein matters very much into causing specific reactions to take place. All right, so we're going to knit together or hook on one amino acid to another amino acid, and they're going to be held together in what's called the peptide bond, okay, to make peptides, which are like small proteins. Just a couple of amino acids together is a peptide, and then many amino acids is a protein. And somewhere in between is a polypeptide. All right, so our proteins will be made also of carbon, hydrogen, and oxygen, must have nitrogen in it, and some of them have sulfur, all right? So essential to a lot of cell structure and to function, especially with function, think of enzymes. Enzymes are proteins that in particular will help bring our reactants together so that they react more quickly, okay? They facilitate the reaction between two reactants. We also have a lot of transporter proteins. These will be proteins that are inserted in that phospholipid bilayer of our cell membranes that will allow a lot of our big molecules that can't get through all those hydrophobic tails. Transporter proteins will allow other substances to move through channels that they make. Proteins also make flagella that aid in movement. Now, when we get it, you know, if you take microbiology, you'll certainly see the proteins there for movement. There's only one cell in human beings that is flagellated, and that is the sperm cell, okay? So that flagella on the sperm is made up of protein. And we'll see if when you take micro, lots of the bacterial toxins in cell structures are also made of proteins. All right, so amino acids have the carboxyl group and the amino group. We looked at one picture of it before, we'll look at it again. And then they always have what's called an R group, which is the side chains, usually chains of hydrocarbons. So here you have, again, your amino acid structure. So here's the amino, NH2, and our carboxyl, the acidic ketone-like carbon double oxygen bond here, and the hydroxyl part, okay? And one carboxyl group will react with the amino group on another amino acid, and it will form a peptide bond between this carbon and that nitrogen, and we will lose water. So right here, we'll lose water. Okay, so here you go, the amino group. Here's the what's called the alpha carbon, the carbon to which the amino and the carboxyl group are attached. If you go back here, this is the alpha carbon. This is the carbon to which the amino group and the carboxyl group are attached, all right? And then you could say, why is it not this carbon as opposed to this carbon? Because you don't see the amino group or the carboxyl group is not attached to this carbon. So this is part of the R group, okay? All right, we have 20 amino acids. Sometimes you see 20, sometimes you see 21, okay? It's no big deal, you don't have to worry about that are found in the proteins that occur in nature, okay? Now, our amino acids can exist in what we call either two stereoisomers. This is getting a little bit complicated, so we're not really going to talk about it here for us because that's a little bit chemistry beyond us. The stereoisomers just mean that they're two chemicals that if you held a mirror up to it, you could see that they're shaped just a little bit differently. This is like being a Rubik's Cube and trying to figure it out, that these two molecules are not exactly the same because the hydrogen is over here on the right side, hydrogen is over here on the left facing side, okay? Just like your two hands are isomers of each other, looking at each other, it's like they're looking at the mirror image, but you couldn't take this hand and superimpose it on this hand and make it be the same. The thumb is always going to be over here, the thumb is going to be over here when they're facing in the same direction. And that too affects their ability to react with each other. All right, so what we're going to do with protein synthesis, I see my battery came out. Hang on, let me plug this. I got to grab the plug. Sorry, I'm trying to stretch cords here across the room. My fly by night operation here. All right, so when we're going to do protein synthesis, the idea is my highly technical language of we're going to hook or knit two amino acids together. We're going to do a synthesis reaction, okay? So it requires a dehydration synthesis. The other word for dehydration synthesis is a condensation reaction, okay? So in either case, we're going to remove water and we're going to create a new macromolecule. So really, I should move this over somehow, these arrows. I don't know why this isn't doing this right. Hang on. Try to get this to get on here and point. Should move this over to say remove water means dehydration, whereas creating a new macromolecule means synthesis, right? Those are the two parts of dehydration and synthesis, also known as condensation reaction. Okay, so you might want to make a note of that. The other thing you call it. And we're going to use the amino acids, okay, the amino group of one amino acid, and we're going to bond it to the carboxyl group of another. And the resulting bond that's formed is called a peptide bond. And then we call the molecule itself between two amino acids a peptide. So here we have an amino acid. This is the amino part, right? This is the acid end. This is that alpha carbon in the center. The acid end right here of one in the amino group are going to dehydrate, remove water, and then we'll have a new bond between this carbon and this nitrogen right here. So if we were drawing it in, you can see right here, we're going to remove all this. So we can draw it in. We're going to form our bond from this carbon to this nitrogen right there. Okay. And we're going to take all of this out of here, right? And then we're going to look like this. NH and NH is the same. HCr, HCr. We have our C double bonded to our O, and then we're going to have our N single bonded to the H. This looks the same. This looks the same. And this right here is the peptide bond. Okay, that's your peptide bond if you needed to identify it, which, you know, sometime like on a test, I might ask you, or PBL, I guess, since that's what we're doing, I might ask you to identify the peptide bond. All right, so our peptide bond, if we look at it this way, we're going to do our dehydration synthesis. Again, we're going to remove the O and the H, and we're going to form the new bond between the carbon and the nitrogen. And it's right here, our peptide bond. This was glycine. This one was alanine. Okay, this is the most simple amino acid, just hydrogens attached to the carbon here, alpha carbon. This has the smallest possible carbon group on it, which is a little methyl group, just one carbon. So glycine and alanine, the two most simple ones, now forms glycyl alanine, a dipeptide, okay? A dipeptide. Then we can have one after another, protein after another, glycine, alanine, methionine, valine, histine, leucine, threonine, you know, going on and on and on with other amino acids, okay? This is the primary structure of the protein, the literal script of the names of the amino acids, one after the other. Met, val, his, leu, 3, okay? This is the next levels of structures, or the secondary structure, how it twists. This would be its tertiary structure, how it twists and wraps around each other from these motifs of the helix, okay? And we can form even more complicated structures. So let's look at the formation and decomposition of a polypeptide. Here, we're going to make dehydration synthesis, also known as condensation. What are we going to be removing? Water, okay? And as we remove water, we form a new type of bond between the carboxyl group of one amino acid to the amino end. That bond is called a peptide bond, okay? And we do it again here between valine and tyrosine. So it made one bond at its nitrogen end. It's making another bond at its carboxyl end with the nitrogen end of the next molecule. So valine to tyrosine. Same thing here from tyrosine to cytosine. Now we're using the carboxyl end of the tyrosine to the nitrogenous end of the cysteine. And so you have multiple bonds, and this is how we form a polypeptide. We've taken away the oxygen and made peptide bonds between all of those. And spend a little time staring at this. This is the reverse taking place. We have this peptide that was down here. We've now moved it up here. We're adding the water back in. We're now going to like split it with water. We're going to break apart the bond, shoot that water in there and have some enzymes to help the reaction along. And now we're back to our original serine, valine, tyrosine, and cytosine. Okay, we've reconstituted this as a COOH end, this as an NH2 end. Our levels of protein structure, again, the amino sequence, like the beads on a necklace, beads on a rosary, like that, one after the other, that sequence is the what we call the primary level of the structure, okay? Then how the structure twists around on itself, whether it might form, you know, pleated sheet, or whether it forms an alpha helix, that's its secondary structure. Then the secondary structure that's kind of like twisted right here might also fold back on a clump of itself. That's its tertiary structure. The quaternary structure is when you have one polypeptide that comes and associates with a whole other polypeptide. They're all tertiary and wound up on each other, and then they come together and form a unique fold into each other. This is what we're going to see when we see like hemoglobin. Hemoglobin is formed of 4 chains, 2 beta chains and 2 alpha chains that come together and make our hemoglobin molecule. So that will be the alpha and the betas coming together to make that molecule. This is usually the 2 places where you have a harder time trying to understand which level it is, okay? Here's another from another book, the same thing, showing it with different pictures, whichever one helps you to understand it, okay? And you need to understand it because protein shape is going to be everything, especially when we get to hormones, cell communication, and enzymes, okay? Structural proteins are going to form the basic shape of our body. Collagen, you'll see like collagen in our body that will line the tips of our bones and sort of be the things that our bones grow down on and form the tendons that help connect our muscles to our bones and all that. A lot of this collagen gives you the supportive structure under your skin. When your skin starts giving out and kind of sagging down with age, you're getting usually collagen depletion in your skin. So it forms a really nice, big, important structural function to our body. And that's because of the shape of collagen. And if you look at collagen as a molecule, it forms like long, long strands of protein, okay? Protein can be denatured. It can be denatured by putting it into a hostile sort of pH for it. The pH in our body, if ever you've had your pH taken of your blood, you get it drawn and they find out what the patient's pH is. We're usually right around 7, a little bit on the basic side. We're really more like 7.35 to 7.4. If you become acidotic, everybody will worry about you and you'll be in the ICU, okay? So you're getting down below a pH of 7, okay? Acidotic. This happens a lot with people who are in DKA, diabetic ketoacidosis, all right? And the problem is you start denaturing the proteins of your own body. You start destroying your own body at hostile pH or hostile temperature. So you're hyperthermic. You fell asleep out in the sun, okay? And you got really too hot. You got so hot, you're beyond sunburn. You've made your body hot, right? You've reset your temperature core. So you're hyperthermic and we need to cool you down. We're going to need to come and bring you on cooling blankets and stuff to cool you down. So you're not continuing to denature your proteins. The ultimate example of denaturing proteins is your egg. An egg is mostly protein, and you take it and throw it on a frying pan and you denature the protein of the egg. And in that case, it is irreversible. You can't get the egg that you just fried after you added heat back to it. You've completely denatured the protein, okay? When you're getting a permanent or when you're straightening your hair and you put all those acidic or basic chemicals on your hair, you're trying to change the natural shape of the protein of the hair, uncurl it or curl it, whichever one you're doing to it. Isn't it crazy that we have both of those things? Those with curly hair aren't happy with it and want to straighten it. Those that have straight hair covet the curly hair. We always want what we haven't got. I don't know why. It's just kind of human nature, isn't it? We're always trying to keep up with the Joneses. It's pathetic. We're pathetic human beings. Okay, we should be happy with who we are. All right, so proteins also will have different parts of them that perform their different functions. So, and we'll see how specific things like what we call the spike protein on our coronavirus will insert on a specific receptor on our cells called the ACE receptor. So the spike protein comes along, the ACE receptor picks it up, and they lock into each other like glue. And this is what our neutralizing antibodies that we make from the vaccine is trying to do. The neutralizing antibodies will come in and they will attach and block to the spike protein on that coronavirus, and it can no longer fit into that tight little area on that ACE receptor. Okay, and that's why we're all trying to get everybody vaccinated, but people can't understand that very well. All right, so you can see proteins here. Here is just what I'm talking about, an immunoglobulin, an antibody. Okay, and it's going to have a specific shape and it's going to bind right on that dang spike, evil spike protein of the coronavirus and prevent it from being able to fit into the receptor that causes it to be taken up by our cells. Here, like I was saying, collagen, which gives us a nice structure to our face and shape to our face. And you can see now, and I'm going to be 57 in November, I'm starting to get some droopiness and loss of collagen, okay, in these cheeks that used to be up here. There's other things in my body that used to be up higher too that are now drooping from the collagen lackage here, but we don't want to get into that. All right. All right. Then we have the gravity's getting to me, right? So we have other things, troponins. When we have our cardiac troponins, okay, this is what kind of triggers our muscle fibers to stretch over each other and kind of tighten up and make your heart beat and pump, okay? DNA polymerase is a protein that's an enzyme, and it fits right on our DNA and helps open up or unzip our DNA so we can polymerize and make new copies of our DNA. So these all have to have very specific shapes to fit on the molecules. And when I see denaturing, like frying an egg, here's the protein with its normal shape. You denature it, you open it up, and you can't really oftentimes, like with the fried egg, you can't go back. On occasion, we're able to recover it and renaturate it back to its normal protein, but not always. Sometimes it's irreversible. All right, so the other thing is our nucleic acids. We're almost getting to the point where it's 7:35. Do you want to take a break now and come back and finish this and start working toward our lab? Okay, so we'll take a break. How long do you want? 10 minutes or 15?
15.